Hello and welcome to this webinar titled Understanding the Propensity of Sequence Variants During Cell Line and Culture Process Development, hosted by Biopharma Asia magazine and presented by Dr. Lisa Marzilli, Associate Research Fellow and Group Leader of Mass Spectrometry at Pfizer, USA. My name is Stephen Edwards and I'll be your moderator. Now, please allow me to introduce our presenter. Currently an Associate Research Fellow, Lisa is a Group Leader in the Mass Spectrometry and Biophysical Characterization Group at Pfizer in Andover, MA. Lisa received a Bachelor of Science degree in Biology from Union College in Cincinnati, New York, and a Master's in Teaching from Boston University. She taught high school chemistry before returning to graduate school for a PhD degree in Analytical Chemistry from Northeastern University, Boston, Massachusetts. Before joining Wyeth, Lisa was a postdoctoral fellow at Johns Hopkins Medical School, Baltimore, MD. She joined the Mass Spectrometry Group of Genetics Institute, Wyeth, in Andover, MA, in 2000 and has continuously worked on the structural characterization of protein therapeutics using mass spectrometry-based techniques. I will now be handing over to our presenter. Welcome, Dr. Marzilli.
Great. Thank you very much. So today I'm happy to present some of our findings and learnings about sequence variants during cell line and culture process development. And before we start, I'd like to acknowledge my co-authors. As mentioned, I'm an analytical chemist, but we're also, this team for sequence variants is leveraging the experience of cell line development and process development from my colleagues here. So Jen, Melissa, Karen, Olga, and Bruno, and Jason, also big contributors for this talk. So to define a sequence variant, it's essentially an unexpected amino acid change in your protein sequence. And the cause, as we're going to talk about today, can either be a genetic mutation in, for example, DNA, or it can happen during transcription or translation as an error, leading to what we would call a misincorporation. And in our talk today, I'll be talking about work that we did with our cell line development group with genetic mutations, and then I'll be talking about misincorporation work that we did with our culture process development group. So here is a picture of a CHO expression system, and essentially you can have a sequence variant occur due to a mutation or mistranslation at any part of that expression system. So, for example, starting with putting your DNA in your plasmid, you can have a mutation in your plasmid before it even goes into the CHO cell. You can have a mutation in the genomic DNA during transcription, during translation. We're also processing and secreting our protein. So there could be an error in any of this biology system. And keeping in mind that we make proteins, we're combining biology and technology. So as Karen Anderson always says, it's biology, so you can have a mutation occur, at any point. So how did we get involved talking about sequence variants? So our first sequence variant that we detected at Pfizer was in December of 2010. And an analytical chemist detected 2 high-level sequence variants in a particular clone of an antibody. And she saw a phenylalanine to tyrosine sequence variant at position 151, which was about 31% in intensity, and threonine to proline at position 304, that was about 38%. So these are 2 very high-level sequence variants. They were confirmed genetically, so we knew they were a genetic mutation, and that was the first time we had ever seen that. This was in preclinical development of the molecule, so we could change out that clone. But if you notice, we started to sort of track what we were seeing, and below is what sequence variants we detected from 2010 to 2014 in our project by clones. So we started looking at all different clones and what we were detecting for levels of either genetic mutations or misincorporations. And you can see there's a number of clones that contained high level of genetic mutations, here abbreviated as GM, and low level. And then really the high level genetic mutations stopped occurring, and we started, but our methods were already in place, so we started picking up misincorporations. So we'll talk about that later. During this, of seeing some of these high level genetic mutations, we put together a team that was cross-functional, cross-site, that could really delve deep into our sequence variant experience, and that team still exists today, and I'll talk about that. But what we did, the first thing that this team did was put in place a sequence variant clone strategy. And the main point of that strategy included having orthogonal methods to screen clones right up front. So this was the first methods we put in place to start screening for sequence variants. So on the top left, our cell line folks did some extensive clonal sequencing. This is the Sanger clonal sequencing method. So they're looking at variants genetically with the sensitivity at that time about 5%. But we wanted to also put in place orthogonal methods to look at the protein. So method number 2 is our LCMS peptide mapping, where we're looking for any new species that may be indicative of a mutation. And to give us a little bit more confidence, we also put in place number 3, the LCMS subunit analysis, which we believe the sensitivity is about 1%. So we're looking for any new species there. And so for all our top 4 to 6 clones that were being considered in development, we would confirm by all 3 of these methods. So therefore, if our cell line analysts detected something by ECS, we could confirm it in the protein, and if someone detected a sequence variant on the protein side, we would confer with our genetic folks. And in general, whenever one person saw it, if it was a genetic sequence, the levels actually matched up pretty well between what was expressed in the protein and what the level of the sequence variant was genetically. So as an example, so here's an example of, for example, all our Lys-C peptide maps when we were screening clones. And as you can see for clone 2C10, there's a new peak that's obvious, so a pretty high level new peak. And therefore, what would happen is we would confirm with our cell line development folks that this is in fact a genetic sequence variant, and we would eliminate that clone going forward. So, the strategy was to screen clones and eliminate whenever we detected a genetic sequence variant. So at the same time, we felt like we needed to understand more about sequence variants. So essentially, trying to understand the root, if we could understand the root cause of why we were seeing these genetic sequence variants. But essentially, those are very long-term experiments to study genetic sequence variants. So and it was going to be resource intensive. So we decided to hire a postdoc that would focus strictly on sequence variants. And her name's Jen Lin. And so she essentially split her time between the cell line, Karen Anderson's cell line development group, and my mass spectrometry group. So she learned methods in both, so she could track sequence variants genetically and also at the protein level. And so what her first goal was to try to understand genetic sequence variant behavior. So how do sequence variants behave under different culture conditions? Do they change? How sensitive to process changes are they? And then the second question is, how do they behave with increasing generational age? So as the cells are doubling in population, does the sequence variant increase or does it decrease? And we chose 4 different cell lines from 4 different antibodies. The top 2 antibodies, antibody 1, antibody 2, had a high level of sequence variance, one at 35% and one at 50%. And then the bottom 2 antibodies were we call low level, under 10% sequence variance, around 7%. And so keep in mind these genetic sequence variants tended to be at random positions, heavy chain, light chain, no specific pattern in terms of where they were located. Okay. So what she found, and her work, by the way, is published in 2017, and the link is on the screen as an attachment if you want more details into her work. But essentially, looking at various culture conditions, different vessels, different scale, different media, essentially she found that whether it's high-level sequence variant or low-level sequence variant, the culture conditions did not have much of an impact on the level. So one representative example is shown below here. So for her work, she looked at the sequence variant in the DNA, RNA, and protein. So she looked at all 3 using different methods. And here showing a bar graph of, on the y-axis, the level of the sequence variant versus all different conditions. In this case, a fed shake versus a 1-liter bioreactor versus a wave bag. And essentially, the sequence variant at about 8% was pretty consistent across all the different conditions. And you see a little bit more in the 1-liter, but overall no dramatic changes. And then the other point here is that she's really seeing good correlation between the sequence variant level in the RNA, in the DNA, and in the protein. So pretty consistent across the board. So that was good news, that our culture conditions weren't essentially trending our sequence variant higher or lower, and that could matter if you had to have a control strategy for that sequence variant, for example. And then the impact of generational age on the sequence variant level was done. So here's an example of our high-level sequence variant. This is a sequence variant that's at about 50%. So here's looking at the level of sequence variant at 10 generations, 50 generations, and 100 generations of that clone. And in general, the level of RNA and protein are very consistent across. So we didn't see that sequence variant growing in population over those generations or decreasing. Those high-level sequence variants pretty much remain constant across the generational age. But you might notice that there's a big difference between the level of sequence variant in the DNA versus the RNA. And in this case, what that tells us is that you have a very different transcription rate for your sequence variant, so that variant copy is making much more of the RNA transcript. And our cell line development folks would call that it's very transcriptionally active. And that was true of both those high-level sequence variants. Same behavior. So the behavior for the low-level sequence variant, this sequence variant is about 7%, 7-8%. What we see here is that from 100 to 50 to, from 10 to 50 to 100, we actually see that sequence variant decreasing with increasing generations. So essentially, that sequence variant population is growing out, if you will. And that was true of both our low-level sequence variants. They both decreased with generations. However, I will say that we've also detected clones that at 10 generations were very, below our detection limit, and that actually increased with generational age. So, I'd say that each sequence variant has a unique behavior profile, and it sort of has influenced our strategy, so that, as you'll see in the next couple slides, our sequence variant strategy is to look at clones at lower generations, cells at low generations, and at high, to see how the sequence variant levels change. So as we progressed in terms of methods, our methods also changed with time. So instead of doing our Lys-C method and our subunit, we developed a method specifically for sequence variant analysis that involved peptide mapping by LC-MS/MS, so a little bit more sophisticated. So in this case, we're taking samples, protein samples that are Pro-A purified. We're reducing, alkylating, and digesting them with trypsin. We're separating the peptides chromatographically, and then they are introduced into a mass spectrometer. And we use a Orbitrap mass spectrometer, so we have both a Fusion and a Lumos thermo instruments that are, really sensitive and fast. And so our mass spectrometer gives us not only the mass of each peptide, but we also fragment each peptide. So we're getting MS/MS data that will allow us to determine the actual site of the sequence variant, which amino acid exactly. And that generates a huge amount of data. So most of the bottleneck comes in the data analysis. So we're identifying peptides with sequence variants, and we're quantifying them. And we're leveraging off software to do that. But it's still a huge amount of data to go through. So, in general, we usually have a month to kind of go through that information of clones. Most of it reducing false positives, because the software will pick up a huge number of false positives. Okay, and on our genetic side of things, our cell line development group now does next generation sequencing. So they're starting with RNA, preparing a library, then amplifying and sequencing, and then aligning and data analysis. And their data analysis also can take a while. So they're in the data analysis is in-house developed Pfizer bioinformatics workflow. So also using software to look for sequence variants. So those are the two methods that we're using for genetic sequence variants. And essentially our current strategy is to screen the top 4 to 6 clones, looking for genetic sequence variants by next generation sequencing. And their sensitivity is approximately at 0.5, so we'd be able to screen out any sequence variants with 0.5% or higher. If there's any sequence variants in that 1% or lower range that they're picking up, we would also confirm analytically at the protein level by peptide mapping, LC-MS/MS, more for understanding the next generation sequencing sensitivity. So any clone that would come up as positive there, we would eliminate it as a possible choice going forward. And then, as I showed earlier, because generational age, we're seeing low-level sequence variants either increase or decrease, we screen by both methods. So we'll screen a lead and 2 backups. We're screening for genetic variants by next-generation sequencing, and orthogonally, we're doing peptide mapping by LC-MS/MS. And here, the LC-MS/MS method has a sensitivity of about 0.1%, so we're reporting anything that we see that's a variant above 0.1%, and then they can confirm that as well genetically. And the ultimate goal is to have a commercially viable clone going forward that does not have detectable sequence variants above 0.1%. So that is our current strategy of screening to eliminate genetic sequence variants up front, so that you can use that same clone all the way until commercial. So it's actually suitable for a commercial situation. Okay, so we've talked about genetic mutations and ensuring that our clones are commercially viable and not containing genetic mutations. So now we're going to switch gears and talk about process-related amino acid misincorporations that may be due to bioreactor process. And so you can also get errors during transcription or translation, as we showed at the beginning, and we call these misincorporations. And one of the distinguishing features of a misincorporation is that it's not just at one amino acid, like a genetic mutation, but it would be at multiple amino acids. So it has some distinguishing features from a protein perspective that you would know is a misincorporation. And these also tend to be very random at which residues they're at within the protein. So we're going to first kind of go over a little bit of how misincorporations occur. And these figures are taken from the Biotechnology Advances paper referenced in the bottom right. This is Ed Wong's paper from Amgen that really has nice pictures of what's going on. So misincorporation can occur due to misacylation. So these are issues during translation. So one is amino acid misrecognition, essentially typically because you're running out of a certain amino acid, you have amino acid starvation. Or you can have tRNA misrecognition, and that could be because you have tRNA scarcity. So in both those situations, you're essentially pulling in the incorrect amino acid. So if you look to the right there, in that top panel, you're pulling in incorrectly a phenylalanine. So even though your codon anticodons are correct for tyrosine, you will see a tyrosine to phenylalanine change or misincorporation. In the bottom portion here, you're incorrectly pulling in a methionine residue. And once again, your codon and your anticodon binding are correct. But instead of arginine, you will see a methionine at different residues in your peptide, in your protein sequence. So in general, we call these mischarging. And the example I'll have today as a case study is a mischarging example due to amino acid starvation, the top mechanism. So in addition, besides mischarging, you can actually have codon-anticodon mispairing. So in this slide, your acylation worked out fine, so you're bringing in the correct amino acid. But in this case, if you notice, your codon and anticodons aren't matching. One's red and one's blue. So in that middle position, the G should match up to a C, consistent to a serine. But in this case, you have a mismatch to a U, and they call that a GU mismatch. And as a result, instead of having a serine in your amino acid sequence, you'll have an asparagine. And in this case, this tends to occur at a low level. It's called GU mismatch, or if it's in the third position, you may have heard it called a wobble mismatch. And ZZ from Amgen did a whole paper discussing this in detail in 2013. So if you want more detail about it, I would totally recommend that paper to discuss mismatch. But it brings up the point that there always is this low level biological noise. And in the paper, they basically say this biological noise is less than 0.1%. So it's impossible to have no sequence variants in your protein, because you definitely will typically have some sort of low level sequence variant due to this codon-anticodon mispairing. Okay, so that's the mechanism. So let's talk about a case study. So one case study showing here today is a Valine Misincorporation example of an antibody that we looked at by peptide mapping, LC-MS, and this is Melissa Lee's work. And essentially she's graphing here the misincorporation at all the different valine amino acid residues in an antibody. So on the left, on the bottom there is different valine residues in the light chain, and on the right bottom is different valine residues in the heavy chain. And on the y-axis is the percent of misincorporation. So the bars represent valine to isoleucine in purple, or valine to methionine in light blue. And what you can see is if you look at that 0.05% level on the left, now we're considering that sort of that biological noise, and at almost every valine peptide containing valine, you can detect a very, very low level of misincorporation. But what we try to detect and read out is any misincorporation that's above 0.1%. So in this case, we definitely have a couple peptides that have more than 0.1% sequence variance. So that would be an action for us to start investigating that. Or if you have cumulative more misincorporation than 0.5%, would also be a trigger for us to start looking at our process. So in this case, we have both, as I'll show you in the next slide. Oh, actually, before we get that, just a couple points of why you would have amino acid depletion in your process. So it could be because you don't have enough supply of that particular amino acid, in this case valine, in your media or feed, or you have a big imbalance of your amino acids. Both can cause misincorporations. So within our groups, our bioprocess folks have started testing the media by amino acid analysis so they can detect amino acid depletion. And then, so that would tell them if we're running out of any particular amino acids, or at least at risk or getting close to running out. And then our analytical group would look at the protein by the same method that we looked at for genetic mutations, peptide mapping by LC-MS. So looking within that protein for any amino acid changes. Okay, so in terms of methods, this is the amino acid analysis workflow that our bioprocess group uses. It's a Waters AccQ-Tag labeling of amino acids. They separate them chromatographically, and then they can integrate the Empower data, that chromatogram, and so that gives them a quantitative method for each amino acid, and using standard curves. So they can get a level of how much amino acid they have in their media. So keeping with our valine story, we looked at multiple batches of this antibody, trying to determine how much valine to isoleucine and valine to methionine misincorporations we were detecting. So in the table on top is the peptide mapping information. And as you can see, the data I showed you originally is from batch A. So Melissa was detecting a cumulative of 1.8% of valine misincorporations. And that was the fed batch that was used. So they started doing some development. So they did batches C, D, and E, where they increased the valine 1.6%. to avoid that misincorporation, and they also increased the nutrient feed rate. And what you can see is in the green, batch C, D, and E, she was detecting about 0.29% misincorporation or 0.4. They did batch C twice. Batch D, we were probably, very, very low amounts of misincorporation, so that looked good. But then batch E was 0.4, so kind of close to that 0.5% limit that we have. So what they did, further optimizing, they tripled the valine delivery for that batch, and that's in blue, batch F. And they also increased the concentration of valine in the nutrient feed. And in that case, if you look at the amino acid analysis, if you follow that blue line, you can see by tripling the valine, you're not running out of that valine amino acid by day 9, that you have a residual amount still left over even after day 12. And in the mass spec side of things, 0.02% was all that could be detected in that protein at any of the valine residues, so super low misincorporations. So that's what we're looking for, basically being able to provide enough amino acids so that we don't have any mischarging, so you're not putting isoleucine in your sequence instead of valine. Okay, so our current strategy at Pfizer is to use amino acid analysis to screen for any amino acid depletion pretty much every small-scale pilot and clinical batch. So we're testing every batch just to verify that we're not running out of any amino acid. So that's our test to understand the risk of a misincorporation. And that's starting right from the beginning of projects, from early all the way through development. And if they do have a risk of running out of an amino acid, then we will check the protein and determine which sites are being modified and at what levels by peptide mapping LC-MS. So once again, our action level is 0.1% at a particular site and 0.5% of a cumulative misincorporation, since it can happen at multiple sites. And we have amino acid supplementation strategies ready to implement. So if something runs out, we can implement them immediately. And I'd say over the years of doing this, pretty much we've been very in tune to making sure that we have enough amino acids in our media now so that we don't actually run out. So it's unusual to run out at this juncture. Okay, so in summary, we've put in place a monitoring strategy for sequence variants. So we're screening with our cell line development team. We're screening for genetic mutations at our original clones with the hope that we eliminate them. So we're not choosing any clones that we can detect a genetic mutation. In terms of misincorporation, we're working with our cell process development group. And while they do amino acid analysis to see if any amino acids have been depleted, we'll look at a few of their top process choices and make sure that we don't also detect any sequence variants due to misincorporations above our action limits. So overall, our goal is to try to minimize those misincorporations or eliminate any genetic variants so that we can make the highest product quality that we can. But also keeping in mind that it's impossible to be 100% sequence variant free because there's always that low level misincorporation at that biological noise level. So our sequence variant team, this is our current sequence variant team, and we've had a bunch of members come on and off of that team shown on the right. These folks are from analytical, cell line development, and process development. And we also serve as a resource for all projects going forward and to keep up to date on methods and literature of sequence variants. And if you would like to know more about our strategy and how it evolved over time, we've recently published our whole comprehensive orthogonal approach to sequence variants. It just came out in MABS in January of this year. So if you'd like more detail about that, that's also an attachment or link on the side of this webinar. Feel free, there's a lot more detail there and contact information if you have questions going forward. So thank you for your attention. And I believe we're going to questions.
Yes, thank you, Dr. Marzilli. We will begin the question and answer segment of the webinar, but once again, I would like to remind our audience that you can still send your questions via the questions tab located directly below your webinar screen. So now our first question is, what is the balance of mass spectrometry versus genetic sequencing for detecting genetic sequence variants?
So the balance of mass spectrometry versus genetic. So in general, we're using next-gen sequencing, which is, in our case, we're using RNA-seq to screen those clones up front. And then mass spectrometry is being pulled in for the late generation clones just to have an orthogonal method and a little bit lower sensitivity. But really our genetic methods do a little bit more there because their throughput is a little bit more. So essentially, it's a little bit balanced toward the genetic sequencing for the clones, and we confirm at the end, at the top and the 2 backups. So, and the reason why we do the top and 2 backups is if we detect a genetic sequence variant, we're going to drop whichever clone that is. So we always, in the end, should have a top clone and a backup clone.
Thank you. Our next question. If only using a mass spectrometry-based method for sequence variants, what gaps do you have?
So every method essentially has gaps. And so our peptide map, our mass spec-based method is a peptide map. We use a Trypsin peptide map, so it's pretty small peptides that we're recovering. But we only typically get 97% coverage with that map, so there's very often some very small peptides that aren't recovered on a C18 column. So if you had a sequence variant, any of those small peptides, that would be a gap. One other place where there'd be a gap is if you had large peptides where you're not fragmenting them very well, then your software wouldn't be able to pick up that sequence variant. So that would be another gap.
Well, thank you for your answers. I think, unfortunately, that's all the time we have for questions. But before I finish the webinar, I'd like to ask our presenter, Dr. Marzilli, if you have any closing remarks.
So I would say that sequence variants are pretty common, and we were surprised to learn that. One thing I didn't mention, but about 20% of our clones have a sequence variant. So I'd say recommending orthogonal methods to detect them early so that you can just eliminate clones is probably the easiest way to go, because it's a lot of work to change a clone later or to have a control strategy if you have a sequence variant later.
Well, thank you. And now I would like to take this opportunity to thank our presenter once more. That was Dr. Marzilli, Associate Research Fellow and Group Leader of Mass Spectrometry at Pfizer, for sharing their knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com. I will get those answers to you. Thank you, everyone. Have a great day.